Topic 6, Human Physiology Part 16, Hormones, Homeostasis, Reproduction, C, is the third of 4 parts on hormones, homeostasis, and reproduction. This movie will have its focus on reproduction, specifically male reproductive anatomy and physiology. The essential idea is hormones are used when signals need to be widely distributed through the body. This slide and the next provide an outline of all the movies available in the Human Physiology Topic 6 unit. Use this outline to find the movie you need for review. The focus of this movie can be seen on the next slide. This movie is focused here. Before getting started looking at the reproductive processes in mammals, humans specifically, it's important to compare and contrast the internal fertilization process that we see in mammals with the external fertilization process found in so many other species. External fertilization, seen here with frogs, requires water to sustain the egg and sperm and requires proper timing in the release of sperm near the eggs out in the open environment. Once the eggs and sperm are released, there is no further parental involvement. External fertilization puts the eggs and embryos at high risk, so females expend substantial energy into making many eggs. The processes of internal fertilization provide inherent protection to the sperm, egg, and ultimately the embryo. Thus, with species like mammals and birds that utilize internal fertilization, fewer eggs are produced as the embryos are protected and substantial parental care is invested in the offspring. Internal fertilization requires complex cooperative behaviors between the male and female to bring the sperm and egg into close proximity, and complex internal organs are required for the protection given to the embryo. In a terrestrial environment that is inherently dry, where the threat of dehydration is high, and water balance is critical to the survival of every individual organism, internal fertilization is quite adaptive. In humans, as representative mammals, the testes and ovaries are the complex internal organs involved in sexual reproduction. As well, the uterus in females is the organ in which the fetus grows. Through meiosis, haploid sperm, haploid eggs are produced, and they are brought into close proximity of each other to accomplish fertilization, resulting in a diploid zygote. The zygote develops into an embryo and then a fetus. Following a 9-month gestation period in humans, the birth of a baby happens. Species that utilize internal fertilization display complex behaviors to accomplish successful sexual reproduction. For example, the display of the male peacock or the necking behavior of giraffes are complex behaviors to ensure successful internal fertilization. Humans have an interesting interplay of biological and cultural behaviors that result in successful sexual reproduction. In addition to the sexual organs where spermatogenesis and oogenesis occur in the male and female respectively, sexual reproduction also involves hormones. Endocrine glands involved in sexual reproduction include the sex organs. The testes produce testosterone, and the ovaries produce estrogen and progesterone. Spermatogenesis and oogenesis are controlled by 2 hormones originating from the anterior pituitary, FSH, follicle-stimulating hormone, and LH, luteinizing hormone. Here are the first 2 IB syllabus statements of importance. Annotate diagrams of the adult male and female reproductive systems to show the names of structures and their functions. State the function of the testes, epididymis, seminal vesicle, and prostate gland in the production of semen. I think it's clear that this movie will have its focus on male anatomy and physiology. Here is an image displaying the reproductive structures in the male. I will start with anatomy, proceeding in an order that starts with sperm formation and follows the route of sperm release. The sperm are formed within the testes here, within tubules known as seminiferous tubules. The sperm, once formed, move to the epididymis where they mature. Once mature, the sperm are moved during ejaculation to the vas deferens and proceed on a circuitous route that brings them past the seminal vesicle. The seminal vesicle releases fluids into the ejaculate. Then the sperm travel into the prostate, into and through the prostate. The prostate gland also releases fluids into the ejaculate. Within the prostate, the vas deferens joins the urethra. The urethra carries urine from the bladder. The sperm are released from the body through the urethra. The urethra is a common duct for both ejaculate and urine. The structures highlighted in red are those that you need to know. Here's a chance to test your knowledge of structure and function. You can stop the movie now or follow along with me. One is the scrotum. The scrotum allows the testes to hang away from the body, so the testes are slightly cooler than the rest of the body. Two is the testes. Sperm formation, spermatogenesis, occurs here within tubules known as seminiferous tubules. Three is the epididymis. The sperm mature here. Four is the vas deferens. The vas deferens serves to move sperm upon ejaculation. Five is the seminal vesicle. The seminal vesicle secretes alkaline fluids into the ejaculate. These fluids have a bit of fructose as well. Six is the prostate. The prostate secretes alkaline fluids into the ejaculate. Seven is the Cowper's gland. Let's not focus on the Cowper's gland. Eight is the urethra. The urethra carries the sperm as they're released from the body. 9 is the penis itself, and 10 is erectile tissue. I will continue to strengthen the functions of structures as the movie proceeds. The male and female reproductive systems are paired systems. There are 2 testes, as there are 2 ovaries. In this image, we can see the 2 testes as well as the circuitous route by which sperm are moved out of the body during ejaculation. Sperm are formed in the testes, they mature in the epididymis, move through the vas deferens, past the seminal vesicles, into the prostate to reach the urethra while being ejaculated from the body. This image nicely shows the ureters that run from the kidneys into the bladder. Here's another chance to practice both name and function of all the structures I've displayed in the earlier slides. You can stop the movie now and practice on your own. With this slide, let's take a closer look at the contents of the ejaculate. Sperm make up 80% of the semen. Seminal vesicles produce 60% of the semen, and the seminal vesicle fluid is alkaline with some sugar. The prostate gland contributes 30% of the semen, also an alkaline fluid with enzymes that activate the sperm. With this slide, we can link together the structures in which the sperm are formed with the ejaculate released upon intercourse. Spermatogenesis happens within tubules within the testes. The tubules are called seminiferous tubules. The sperm mature over 70 days in the epididymis, seen here as more tubules, all of which converge to become the sperm duct, the vas deferens. 2 to 5 milliliters of ejaculate contains 240 million cells and has a pH of approximately 7.5. This is a micrograph of a seminiferous tubule within the testes. Spermatogenesis occurs in the seminiferous tubules of the testes. Meiosis starts with a diploid cell on the outer edge of the tubule and proceeds in 2 divisions in this direction, resulting in a haploid gamete, an immature spermatid, in the tubule from which it can be released. You can see the flagella of sperm here. The form-function relationship of the 2 meiotic divisions proceeding toward the lumen of the seminiferous tubule is beautiful. All of the cells seen within the seminiferous tubule are germinal, in the sense that meiosis is resulting in gametes. Before looking closely at the details of sperm formation, spermatogenesis, Let's look at the hormonal control of spermatogenesis. Here's an IB syllabus statement. State that a gene on the Y chromosome causes embryonic gonads to develop as testes and secrete testosterone. Here is a karyogram. You should recall the sex chromosomes that determine gender in humans. There is a gene on the Y chromosome that causes embryonic gonads to develop as testes and secrete testosterone. In a developing embryo, seen here, there's a region known as the genital ridge, seen here in this image. If the embryo carries a Y chromosome, the embryo will be male as gonads develop into testes and secrete testosterone. So that brings us to this IB syllabus statement. State that testosterone causes the prenatal development of male genitalia, spermatogenesis, and the development of secondary sex characteristics during puberty. In this image, below the hypothalamus is the anterior pituitary, which releases FSH and LH in the male. Both of these hormones target the testes. Seen here is the entirety of this green box. FSH and LH target the seminiferous tubules directly to stimulate spermatogenesis and target nutritive cells known as Sertoli cells. LH also targets a cluster of cells within the testes known as the interstitial cells of Leydig. The interstitial cells of Leydig manufacture testosterone, and testosterone stimulates spermatogenesis. Testosterone also feeds back to the hypothalamus to slow the release of FSH and LH. This is negative feedback control. Here is a slide that provides the functions of FSH and LH. FSH targets the seminiferous tubule within the testes to stimulate spermatogenesis and stimulate the Sertoli cells, which are nutritive, supporting spermatogenesis. LH targets the interstitial cells of Leydig that secrete testosterone. Testosterone has various target locations throughout the body, but has 4 important roles. Listen carefully. Testosterone is important in the prenatal development of male genitalia. It's important in the development of secondary sexual characteristics such as facial hair, pubic hair, muscle and skeletal development, and a deepening of the voice. Testosterone stimulates spermatogenesis by targeting the germinal cells of the seminiferous tubules. And lastly, testosterone is responsible for the maintenance of sex drive. Here are the functions of testosterone. You can stop the movie now and take notes on your own. Interestingly, many of the hormones in sexual reproduction are steroid hormones derived from cholesterol. We can clearly see the relationship between cholesterol and progesterone, estrogen, and testosterone. By the way, FSH and LH are both polypeptides. They are protein hormones, not steroid hormones. Here's the relevant IB syllabus statement. Lists 3 roles of testosterone in males, including prenatal development of male genitalia, spermatogenesis, and the development of secondary sexual characteristics. Here are the roles. I'll let you stop the movie now and take some notes. So let's now dive into the details of spermatogenesis. Spermatogenesis occurs in the seminiferous tubules of the testes. Here is a micrograph of a seminiferous tubule. Spermatogenesis occurs in the seminiferous tubule of the testes. Meiosis starts with a diploid cell on the outer edge of the tubule, and that cell is known as a spermatogonium. All of the cells seen as a part of the broad wall of the tubule are germinal, in the sense that meiosis is resulting in gametes. The 2 divisions of meiosis occur in this direction. You can see cells, all of which are at some stage of meiosis, that results in haploid gametes, immature spermatids, in the tubule from which they can be released. You can see the flagella of sperm here. The form-function relationship of the 2 meiotic divisions proceeding toward the lumen of the seminiferous tubule is beautiful. The interstitial cells of Leydig would be located here and here between the tubules. In the micrograph on the left, The germinal epithelium is along the outer edge of the seminiferous tubule, and the germinal epithelium produces cells that will ultimately undergo meiosis. And this is germinal in the sense that meiosis is resulting in gametes. The cells undergoing meiosis are here in purple, meiosis proceeding in this direction toward the lumen of the seminiferous tubule, within which we can see the flagella of the sperm cells. On the right, a bit more magnified, again, we can see cells in purple and at various stages of the meiotic process, as well as Sertoli cells seen here in green. Sertoli cells provide nutrition to the developing spermatids. Spermatogonia here are diploid cells that initially undergo mitosis to form two cells, both diploid. Of the two cells resulting from mitosis, one remains back as a true germinal cell, and one proceeds into meiosis. The mitotic step allows for an unending supply of cells that would proceed through meiosis. Without the mitotic step, the germinal epithelium would be exhausted of cells soon enough. Here is a nice diagram showing the relationship of seminiferous tubules to the interstitial cells of Leydig. The interstitial cells of Leydig lie between the tubules. The presence of capillaries makes sense, as the interstitial cells of Leydig are endocrine. Testosterone is released into the bloodstream. You can see the direction of meiosis here as a part of spermatogenesis, and you can see the flagella of developing spermatids here. This is a nice diagram because of the detail. The germinal epithelium would be this outer layer of cells. Diploid cells, known as spermatogonia, are here, and we can see cells undergoing divisions here and here and here. Mitosis, then meiosis proceeding in this direction. This is spermatogenesis. In the lumen, we can see immature spermatids, not yet spermatozoa. Spermatids must differentiate into spermatozoa. This is known as cell differentiation. Nutritive Sertoli cells, seen here and here, with their developing spermatocytes nestled into the membrane. And between the tubules, we can see the interstitial cells of Leydig that secrete testosterone. And this IB syllabus statement will bring this movie to a close. Developments in scientific research follow improvements in apparatus or technology. William Harvey failed to solve the mystery of sexual reproduction because effective microscopes were not available, so the fusion of gametes and subsequent embryo development remain undiscovered. William Harvey, well known for his work on blood circulation through the body, also studied mammalian reproduction, specifically in deer. Harvey also established several conclusions surrounding theories of reproduction. First, he denounced spontaneous generation by claiming that even maggots and worms have some origin in eggs. Harvey rejected theories from Aristotle about early embryogenesis involving embryo formation from the combination of menstrual blood and semen. Harvey did provide new information about conception and early human development, but he could not confirm many of his theories because he could not see single cells. It took the invention of the microscope 17 years after Harvey's death to confirm conception and early embryo development. The microscope was invented 17 years after William Harvey's death, so William Harvey failed to solve the mystery of sexual reproduction because effective microscopes were not available, so the fusion of gametes and subsequent embryo development remained undiscovered. And that brings us to the end of IB Bio, Topic 6, Human Physiology, Part 16, Hormones, Homeostasis, Reproduction, C.